The first description of the kingdom Protista was given in 1676 when Anton van Leeuwenhoek invented the first microscope whose magnification was in the range of 70x to 250x. And here one thing is that the present day light microscope, whichever you are using in lab, the magnification is around 1000 times higher. And when we go for electron microscope, it could be like several lakhs and all. So anyway, with the discovery of the first microscope itself, it got a hint of the first groups of animals or whatever organisms, and hence they have classified it as Protista, meaning protos means the first and zoon means the animals. However, in 1862, a German biologist named Ernest Haeckel was the one who described these groups as they are neither which could not be clearly distinguished as an animal or a plant cell, and later it was identified that it comprises of several phyla, and hence it is a polyphyletic taxa, means it is not descendant from a one ancestor. It has several ancestors. For example, ancestor in the sense we know that humans, the ancestor could be traced back as monkeys and all. But here it is like for them, for the protist, it have several ancestors are there. And the interesting part of the Ernest Haeckel work is that since there was no instrument to photograph this images capture seen in the microscope, he used to draw it and color it, and with that we were able to identify, or we have to study how it looks. So coming to the characteristics, most of the Protista unicellular, though multicellular also exist, and some are colonial. So these are the most importantly, they are eukaryotic. So here eukaryotic in the sense that they have a true nucleus and other membrane-bound organelles. So these are the structures of the majority of the unicellular protist, and Volvox is an example for the colonial protist. And multicellular algae could be seen very big, like it can extend up to many meters. So from here itself you can identify that they exist in different sizes ranging from microscopic to several meter in length. Then coming to its habitat, it could be found in variety of habitats, including the oceans, the freshwater, the terrestrial habitat, and even many of these protozoa and kingdom Protista groups are actually parasitic to humans. So even inside the humans or many other similar organisms, they can survive. So it has a wide range of habitat. Then coming to the cell structure, locomotion and osmoregulation. As I said, these organisms are typical eukaryotes because of the true nucleus, and they have their own mitochondria, the energy currency, whatever you call, and also majority of them have this chloroplast and hence capable of photosynthesis. So in this protist itself, they are some groups are autotrophic while others are autotrophic, means synthesizing their own food with the help of photosynthesis, and many others are heterotrophs, and within the heterotrophs many are parasites, and like that, so on. So that is about the structure. And coming to the locomotion, generally they possess three different types of cell structure for locomotion: flagellum, And then cilia. Cilias are the one which is shown in this paramecium, and with the help of that, how it moves, you can clearly distinguish it. And also in amoeba, it is like we know that the amoeba is not having any typical structure, and they can change their shape constantly. Hence, with this pseudopodia, actually they move and also they take the food. And the next method for this kingdom Protista is that they have the ability of osmoregulation. So you have already learned that normally if a cell is placed in a normal solution, it should remain intact like this. So in that condition, for animal cell we call as normal cell, and in plants it is flaccid. And similar way, for example, if water is entering into it, for example, if you see that that dried grapes and or if it is put in a water, the water will definitely enter into it, and it become like bulged. Similar way, in sometimes if you have placed like in that salt mango, when the based upon the concentration, the water may move from that mango to the surrounding, causing it to shrink. So this is the way which actually affects that if there is a difference in the solute concentration outside. So osmoregulation is a mechanism by which, with the help of contractile vacuole, the organism will maintain a normal condition inside by either taking water in or giving out water. Then is about the reproduction. In reproduction, both the sexual as well as the asexual mode is identified, and in the general asexual mode, the most common is the binary fission, where the cell divides into equally two halves. So even the plant cell and also it is very common. And next is the multiple fission, in which one of the nucleus itself, it will divide into several, and each nucleus cells is capable to form a new daughter cells. And then comes the sexual modes, in which one of the most common and the one which you are going to learn is the conjugation, in which it's a method of cross fertilization where the donor cells are transferred into a this recipient cell with the help of a conjugation canal. Then comes different types like autogamy, cytogamy, etc., which is also a mode of self-fertilization where and it happens in the nuclear level. So here just the cell divides and then it distributes and forms their own nucleus. However, in this autogamy and cytogamy also the same cell only divides. However, that division will start with a nuclear organization and followed by endomixis. It's also like similar to that, but however there's a multiple copies may form from a single this thing. So then is and we know that these protozoans can causes many diseases also. So maybe in the next class we will be dealing with more with the parasitic protozoan. So for today I have enlisted three groups: Heliosova, Dinoflagellata, and the opalnids. So here coming to the Heliosova. Heliosova are also a polyphyletic assemblages, meaning they are not originated from one single ancestor. They have many ancestors, means many different types exist. However, they have a similar lifestyle and body forms. Thinking to classify it that as a single phylum, but now it is very clear that it is not a single phylum. Okay, because a phylum characteristics are that it should be derived from a common this thing. Whatever is classified under the phylum should be derived from a common ancestor. But in Heliosova it is not the case. However, having a similar lifestyle, they have a similar body forms. So majority is having a circular structure, and it possesses lot of spike-like projections known as axopodium. And actually for like other cells it is a difference, because I earlier said that cilia, flagella and pseudopodia are the most common. However, in this sorts are lacking, and axopodium is the one which will provide some flexibility or the locomotion ability. Then typically different genera has been identified, which is enlisted here, and one of the type species is the one which will possess all the characteristics described for the given phyla, are this Actinosphaerium, and the most different species are which is denoted by a capital letter followed by the species name here, and as I said, it should be represented in italics. So in which this Actinophilus sol is the most dominant group identified, and here and this type of Heliosova are planktonic in the nature. Planktonic in the sense that they don't have, as I said, they don't have this typical locomotory organ. So their movement is dependent upon the water currents. Wherever the water currents go, this organism can't resist that. It means for example, we have a will to go wherever we like. But for the planktonic groups, they will flow according to the water currents only, and they are found in both the fresh water and the brackish water. And the locomotion, though there is, but it is very less, and it requires that stiff axopodia. And economic importance is that they are most abundant during the warmer month, and they feed on a ciliated protozoan. So definitely, especially, and means during this period, the count of the ciliated protozoa reduces, and when with increase in the count of this groups, hence it is very important in shaping their population and distribution. And what is the general purpose of this axopodia is mainly used for capturing prey, and this also sought some protection from the predators. And these groups generally, as I said, it mainly feed upon many ciliated protozoans. However, some small flagellates and microscopic algae also they feed. Then coming to this phylum Dinoflagellata, also known as Pyrophyta. So here why phylum status is given, because whatever different types of dinoflagellates shown here has originated from a common ancestor. And here, unlike that the previous phyla which is commonly found, this Heliozoa which are commonly restricted to fresh water and brackish water, majority of the phylum Dinoflagellates could be seen in marine habitat. However, recently some groups also exist, but it's most prominent in the marine environment. And why that Dinoflagellata name came, because it has two flagella, and that are actually not equal size. They have different sizes flagella, and majority of this contains the chloroplast, meaning they could be like having a very importance as a primary producer. And one of the dinoflagellate which is most common to majority of your will be like, you must have seen this red tide phenomena in the sea, and this represents the dinoflagellate bloom. And at night, since they are bioluminescent in the sense that they can produce some fluorescence like chemical, at night you can see this type of shining starry colors in the oceans, or even sometimes in the brackish area where salt, based upon the salt content. So this type of fluorescence could be seen. And this is the general typical structure. And here the economic importance mainly relies upon the formation of this red tide or the bioluminescent phenomenon. Then comes the opalins. Opalins are the one, again it can't be classified, it's a single phyla, It has uncertain phylogenetic affiliation, and this could be found in larger number in especially in anurans, that is amphibians, and they often exist as a symbiont, meaning if means in the large intestine, because we know that digestion process and all is taken up by the intestinal this thing, and these groups especially they form a commensal relationship, and they in turn provide it benefited by host by getting the required nutrients and without causing any damage. So unlike the other fission whichever I showed, here the type of fission is mainly by binary fission only. However, it happens in the longitudinal plane, and earlier picture you remember it was through this latitudinal plane. And they have a typical, since it is surrounded by lot of cilias, and they have a typical metachronal wave like that movement. And here the cell surface also bears some supported by the microtubules. This, and one of the other characteristics which is very prominent is that it has this multiple nuclei. That is multiple nuclei. Generally, majority has either one nucleus, and in case of this Paramecium and all, you can see two types of nucleus, and while in the opalinids it is like multiple nucleus is identified. So here, and this is dissolves. So one of the reason economically how they are spreading from one tadpole to other is that during their reproductive stages, during the spring season especially, when the frog starts breeding, the symbiotes divide rapidly and produces many pre-cystic forms. And when this frog get eliminated through frog's feces, and from there it will go into different types of cells. And generally for opalinids, the type of nutrition they follow is like similar to this, the pinocytosis, where the contents or the absorbed solutes are taken by the cell through this process. Okay, thank you so much. This is all for today.